The next point is there are some situations where molecular phylogenetic analysis of any kind may not be the approach you're looking for. There are lots of other methods, and you've used some of them. Let's talk about a few of those. One of them is DNA hybridization. Any of you guys ever looked at Bergey's manual? This is kind of a manual. This is the definitive definition of most bacterial species. And one of the things that's in there is always the DNA hybridization experiment that shows you that your organism is different than its relatives. Typically, you're expected to be measurable hybridization if you're in the same genus, and it's got to be less than 70% identity, or else you're considered to be in the same species. This is a terrible measure by any definition. Let me give you a quick example. If you take E. coli 0157 and wild-type E. coli, say W3110, and you do a DNA hybridization, if you label the wild-type DNA and hybridize with the 0157, they'll be 99% identical and look like the same strain. If you label the pathogen and use the wild-type to hybridize to, they're only 70% identical. How can this be? How can the experiment give you a different number measured one way with one probe and one target and vice versa? 01757 has more DNA. It's got big chunks of added DNA that don't hybridize to wild type. These are required horizontally. And so there, depending on how you do the measurement, you get a completely different answer. And the reality is, too, this is a bunch of work, and so people don't usually do it. Another old-fashioned method is base composition. And so bacterial DNAs typically range from something like 45 to 60% GC. This is a really easy measurement to make. There are two good ways to do it. One is to label the cells with radioactive phosphorus, usually P32. They take that up, they put it into their DNA, and then you extract DNA and hydrolyze it chemically, and then just run the 4 bases out on some kind of chromatography and measure the relative amount of radioactivity in each of the 4 spots. That can be done with exquisite precision, you'd be amazed. A little bit easier and not so radioactive way to do it is to do a melting curve, because the more GC content there is, the higher the melting point. And so you can measure this straightforwardly. It turns out nobody uses this anymore. It's a great way to prove that 2 organisms aren't exactly the same, but otherwise it's not very meaningful. Serology. You guys probably did this in mid-micro lab, right? And if any of you have ever thought you had strep throat, you had a strep test, and that's a serological test. The notion here is that organisms have on their surface a variety of proteins, and you can probe for those proteins with antibodies. Antibodies made against them in various ways. And different strains of the same species often have different surface proteins. That's why they're different, because that's what the host immune system looks for, and so they vary them. And so there are a variety of ELISA tests and so forth that you use to test for different kinds of organisms. Now, there's a big shortcoming to this. If you picked one of the colonies on your plate, for example, you would have no idea what kind of antibody to start with, right? Because it could be anything. Likewise, it's pretty unlikely to be a pathogen, and so probably those antibodies aren't even available. In general, serological tests are best for pathogens, because that's where these banks of serological reagents are available. So it's great for detecting strep with a rapid test. It's great for identifying what strain of salmonella you might have, or what virus. A lot of times these are used for viruses because you can't grow colonies of virus on a plate. And so you look for the presence of antibody against that virus. And there are lots and lots of variations on these tests. Another one is lipid profiling. So if you take an organism, you take a colony on a plate, grow it in very highly controlled conditions, and then take that colony and extract it with octane. Typically, you do a step in the middle that converts the fatty acids into methyl esters. And then run this in a gas chromatograph, you might get a profile that looks something like this. Each one of these is a fatty acid side chain on a membrane lipid. And it turns out that the resolution of this method is really, really high. And with the right standards, you can identify exactly what each one of these fatty acids is. This is 16O, that means it's a 16-carbon fatty acid. That's oleic acid or something like that. I can't remember for sure. 18-1, that means there's one unsaturation in it. There's a 10-methyl, and so forth and so on. And so the fatty acid profile of an organism is readily identified. You've just got this giant database of fatty acid profiles, and if you've got a colony on a plate, you just have the computer look up a match, and there you have it. And this is a really fast approach. Once you have a colony, you can do this whole test in 1 hour. That's pretty cool. Problem is, it can only look for organisms you already know about, right? Because you have to, if you get a pattern that doesn't match anything else, then you don't know what it is. It's also critical that you grow them under exactly the same conditions that the standards were done in. Because organisms modulate the relative amounts and the kinds of fatty acids they use based on growth conditions. The obvious one here is temperature. E. coli has a different membrane lipid composition if you grow it at 30 degrees than if you grow it at 37, because it has to maintain the proper membrane fluidity for that temperature. And the result is you get a different pattern depending on what temperature you grow it at. Likewise, if you put some organic solvent in, if you put in maybe 0.5% ethanol, it's got to adjust for that by changing the membrane lipids. And the result is it can be hard to identify. There's a really obvious case where this has been used a lot, because these are methyl esters, because they started out as fatty acids. But archaea don't use fatty acids. They use alcohols, fatty alcohols. And so if you take a complete population of microbes and do the same analysis, you get one set of peaks for alcohols and one set of peaks for acids, and that tells you exactly the relative ratio of archaea to bacteria in the environment. It's a cool trick. This is being used some in the medical field. It is theoretically possible to look at a mixture of organisms, and you get a very complicated pattern then, right? But computers are really good at picking through patterns and deconvoluting them, separating them out into individual patterns that you can identify the individual patterns from. This method has been pushed really hard recently, but it hasn't gone as far as I would like, I think. Another method are RFLP methods. RFLP stands for restriction fragment linked polymorphism. The reality is almost nobody uses restriction fragments for this anymore, usually PCR products or probes. If any of you have ever had a paternity test done, this is how it works. I'm going to hope not. If any of you have ever been the subject of a forensic DNA analysis, this is how it works. This is used a lot in the eukaryote world. There is huge money in this for example, studying in animal husbandry to make sure you're breeding the right animals for optimum whatever production. This is a great one. This is a set of strains, isolates of Clostridium. These are ostensibly the same species, Clostridium botulinum. All of them serotype 1. Look at the difference in patterns in these things. Compare, for example, this one and this one. I mean, the patterns are really widely different. How do they generate these? These are generated by using random primers, typically short primers, maybe 6-mers, that will amplify all kinds of weird stuff around the genome. And if you do it in controlled conditions, you get a specific set of patterns based on the haphazard location of where these primers can land in the genome. And those will be different in different strains depending on how the genome is changed between them. What's really interesting is that when you measure and you can do trees based on these patterns. This is digital information. You can study it the same way you do other kinds of trees. Really interestingly, if you're looking then at kind of the gross genomic structure, which is what this is doing, and you compare that to serotype, you don't see a lot of consistency. And so what strain you have usually depends on how you measure it. This is a type A, this is a type A, but their genomes are completely different. The serotype has to do with one or two proteins, right? This is looking at the genome itself. Yeah?
So, in what application would you want to look at serotype versus looking actually at the genome and how evolutionary it is?
Yeah, so it depends on what resolution you need and why you're looking. For pathogens, serotype is very often the most important because, again, that's what the immune system is saying. In terms of the genomic rearrangement, you're looking more at kind of the evolutionary history of the organism, who's related to it.
Wouldn't serotype be faster?
Serotype would be faster. If this looks like a lot of work, it is. And not just to get pretty gels like that. I mean, can you imagine how many gels they had to run to get one that looked like that? And then, doing tracings of all these, digitizing them, sorting out the data that you need in order to generate the trees. This represents a fair bit of work. And the point of this paper, by the way, was that the serotype and genotype of botulinum strains don't match. Here's the number one method, though. If you've got an organism, what did you do in General Micro for your unknown? You didn't do ribosomal analysis. You didn't do serotypes or FLP experiments. You tested a bunch of phenotypes to see what it was. And so you can do this a lot of ways. Nobody uses those giant 20 ml tubes of colored fluids anymore. No. The big tubes. Those of you who did the 352 instead of 354 use these a lot. Does Dr. King use the big tubes for anything anymore? Or are they all narrow tubes? In food micro, yeah, that's. And so you can test a bunch of markers to see what sugars it can ferment, what amino acids it can grow on, whether it generates sulfide, whether it can grow on mannitol, etc. Usually these are done in things like interotubes where you do a bunch of tests all at once. This is an API strip. I show this because bioMaroe is nearby during Durham, I think. Where you inoculate a whole string of them, and then it gives you a pattern that you can use and look it up on the chart. The best ones these days are in microtiter plates, so you do 96 tests all at once and read them on a scanner, right? And the computer just pops out an answer. It's beautiful. Yeah?
Do the same situation here where like the phenotype and the genotype can be different? And
Yes. And so, for example, this is the API 20E. This is used to test enterics. You have to already know that it's an enteric for you to find this test useful. You might test some weird gram-positive organism that might give you the same result on this test, even though it's not the same species as E. coli. This particular one's E. coli. So you have to have it narrowed down to the right kind of organism before you even start the test. It's also possible to be fooled by this. If any of you have ever worked with E. coli in a lab, a lot of them are genetically modified in a variety of ways, right? Most lab strains of E. coli are lac negative. But that's one of the definitive identifications of E. coli on these tests. And so if you put a lab strain of E. coli in this, it will probably be wrong. Interesting question. If you take a strain of E. coli and you make it streptomycin resistant, and you nuke the lac gene, and maybe you changed a couple of other things, is it still the same species? I I think the answer to that is your opinion. I don't think there's a rational definition for whether that's true or not. Anyway, you just grow them up, you see what colors they are, and you match the pattern, and you've got your identification. This is the most commonly used for bacterial identification because it's easy. And because usually you don't care so much about the label, what you care about is what it does. Many of these days will also include a bunch of antibiotics, right, to see whether it's resistant to this or that. All right. I'm going to stop this.